Okay, so let's start the next section. It's a great pleasure to have with us today Tnaja Kostic from Austrian Institute of Technology that is going to talk about the world scenario of microbiome for a sustainable food production. Tnaja is a senior scientist at Australian Institute of Technology. She had a BS in microbiology at Carl Simmons University in Austria and a PhD at the University of Natural Resources and Applied Life Science. Her major research focuses in developing tools for molecular analysis to study environmental microbiology. Tnaja, thank you again very much, and the floor is yours.
Thank you, and good morning, everyone. I guess it's still morning at your side. I would like to start by thanking Professor Arruda for giving me this opportunity, but also for giving me this title. I think at the beginning I was a little bit wowed by it, but actually I think it's a really suitable title, The World Scenario of Microbiome for Sustainable Food Production. I think this is exactly what we need, and I hope that in the next 40 minutes or so, I will also be able to convince you about it and show you, what we still need to do to go down this way. In my presentation, I've been mostly presenting the work from two great European projects. On the one hand, the microbiome support, and on the other, the master. You see the two logos and two websites. You will also see from which project I'm reporting based on the slide template. So let's start and see how this story develops. Oh yeah. So first of all, I will tell you a little bit about microbiome support. Microbiome support is a project where is set out to pave the way towards coordinated microbiome research and innovation activities in the food system to support, first and foremost, of course, the European Union, because it's a European Union project, but also international bioeconomy goals. And something really specific about this project, it is not a research project. So in this part of my presentation, you're not going to be seeing any research data. It was a so-called, or it is still a so-called coordination and support action, which has a major aim to sort of investigate the framework's condition and make a recommendation how framework in this setting for the research and development should look like. The project started in November 2018, and we are almost at its end. So it will end, unfortunately, and I say this really unfortunately because it's such a great project and such a great group of people we collected in October this year. However, there's still some stuff coming, so I hope that I will manage to raise your interest and that you will start following us, at least in our last few months. The project was coordinated by my boss, Angela Sesic, at the Austrian Institute of Technology, with the great support of our coordinator, Emmanuel Magin at Entre. What's really specific about the microbiome support, it is an EU-funded project, but also the international partners were funded. So it is a consortium of 27 partners from the EU and so-called IBF countries, and I will come to IBF in a few more slides, so be patient with me on that. And we also have two associated international partners from US and New Zealand. And if you see this world map, we are really, really international on this map. There are unfortunately still some quite gray areas, but we hope that in the future we will also be able to change that. Another specific of this project was that we had three great advisory groups that involved different stakeholders in the project. So we had really active involvement of the representatives from funding and policy sector, from the industry sector, and what we called coordinators platform. Those were the coordinator of past and ongoing research projects that were addressing microbiomes in the food system. We also developed quite a huge expert pool. You will see we had a lot of specific workshops addressing some technical issues. And for those, we of course wanted to have a consensus for the community and always try to involve as many experts as we could. Another specific of the project is we had a lot of exchange with different stakeholders. I already mentioned industry. I also mentioned government and sort of funders and policymakers. But we also wanted to have a good exchange with public. We also wanted to have an exchange with education sector and the science sector. And for that purpose, we sort of implemented an approach of having our partners, our 27 partners, as national ambassadors, who were sort of a link between the project and the consortium and all of the different stakeholders in their country, and who were really facilitating that the different stakeholders participate in different activities of the project, which was really, really great and helpful to achieving our goals. So when we set on this journey 4 years ago, we had 6 major objectives, and I mean, some of them were definitely defined by the call and what the Commission also expected of us. And first was to identify and map what are the microbiome activities in the EU and the worldwide, what are the big programs, what are the facilities, who is doing projects, and in which sectors are projects done. Then another, as I said, really major objectives was really to create a platform for scientists, for regulatory experts, for industry, for policymakers to exchange and to start talking to each other and to see, you know, what are the needs and of different sectors and how can we collaborate to fulfill this aim. Another big issue, I think everyone who works in the microbiome sector knows that there's so much data out there, but how to use the data from before, how to make our data usable for people who want to use it in 1 or 2 years. Can we somehow, are there any standards that could be implemented in this field that is sort of still quite new, is technology driven, is fast paced? But the question is, can we improve something that, because a lot of money is invested in creating data, and it would be to benefit of all if we could use this data. We also wanted to define or try to define how the research and how this landscape should look into the future to really enable us to transfer the applications from the research labs into the application, into the food sector, into industry, to the farmers, and how this path could look like. And of course, there was the vision that I will explain to you about this International Bioeconomy Forum and what it is and what the working groups are, but also to support their work. And this is what we've done in the past 4 years. Somewhere along the way, we also, as a consortium, have sort of formulated this vision, like what do we really want to see? Of course, we want to do all of this, but what is really the big vision? And we said our vision is to really see these products and applications that are in development, these great ideas that, you know, all scientists all over the world have, and not only academic scientists, also industrial scientists, to really see them come into the use and to support this transition into the future-proof food system that will be in line with what we in Europe call Food 2030 priorities, but what you will also recognize in, for example, the aims of the SDGs that are more international. So this was really a vision to say, okay, let's go from this great research and let's go and use the results, and let's change the way how we live and how we produce the food. A nice thing that happened in Europe some time ago is that we started thinking more in the systems approach. So we, you know, like 10 years back, everything was in silos. There was, you know, food primary production plant scientists that never spoke with the food production scientists, and nobody was talking with health people. And then the European Commission introduced this food systems concept, and I find this is really nice, and this is really also in line with the One Health approach, if you think more globally. But European Commission defines food systems like the way the food is produced and how this affects our health, our well-being, and the environment. And you see all of this sort of circular system here in this nice graphic. And I mean, yes, we are all aware that microbiomes will perhaps not play a big role in the logistics, but they will definitely play a big role in the food production. They will play a big role in the food processing. They can also play a role in the packaging, but then especially when we talk about healthy people, about the waste streams, these are all areas where we can think microbiomes. And this food systems is also in the center. I already mentioned some of those in some of the major policy, and I think that sustainable developmental goals are nothing new to anyone in this audience. But if we talk zero hunger, if we talk the use of land, of water, if we think about sustainability, this is also where we have to think about the food systems. And recently, Europe, we also established the Green Deal, and there is also the farm to fork strategy coming again, the zero pollution concept coming again. And we really, when we think about these goals, we need to also think back to food systems. And why I'm emphasizing this so much is this slide, and I took it from one of the older presentations from the European Commission. It was from 2017. But I really like this slide because it shows the perfect storm, as they call it, that we do have in the food system. And the numbers of this slide are actually a little bit scary. We all know that we will have to feed more people in the next year. We also say we will need more food for that. And I'm currently actually attending a phytobiomes conference that is all about producing more food. But I always try to tell, and what I think we should also all think about, we waste a lot of food. And the number here, on the bottom left, the 33% of the food that is wasted, this is actually quite a conservative estimation. There are some estimations out there that are even higher. And this is a terrible number if we consider, we have growing population, we need more to produce more food, we are hitting the planetary boundaries, but then again, on the other side, we are wasting quite a lot. And we see how much energy and how much, especially water and other resources of food production is using, like 70% of water, 30% of energy at the same time, producing quite a lot of greenhouse gases. We have all of these climate goals, food waste goals, zero hunger goals, and yet we are not addressing, or haven't been at definitely 2017, not been addressing the food systems in a systemic approach. And this is what it has to change, because we need to calm this perfect storm down. This is just, we cannot live in this way anymore. This is just not justifiable, and this won't last. Another nice development, and this is not only on the European level, but also more globally, is that the microbiomes have started to gain their recognition in the policy sector. So when we started this project, the microbiomes were in very few policies around the world. They were sort of there indirectly, but they were never mentioned as microbiomes. And in the recent years, on the different levels, we really have them highlighted. Like as you see on the right side, the FAO put out this great little booklet, and I really like it, and I cite it often, asking, is microbiome the missing link towards the One Health concept that we have? And in European Union, the Food 2030 pathway is one of the 10 pathways that were established and developed by scientists, co-developed by scientists and policymakers, was actually putting microbiomes and said microbiome is an opportunity for our society and for our planet. It is one of the 10 really important ways we should go in the future in order to achieve all of these goals that we set ourselves and that are quite necessary. So this is really on the good news that we have seen some really, really positive developments in the past years. And I mean, I don't know exactly who everything is sitting in the room, but I hope that you will find this just, nodding your heads at these slides. And this is a graphic that we have developed within the project. Why are microbiomes important? Because they're all over the place. They're in any environment which you can encounter. And they really sort of play many important roles. And yes, we do have pathogens. We have animal pathogens and plant pathogens, and the ones that are transferred through food and other matrices to the human. But we also have all of these beneficial microorganisms that produce oxygens, that fix nitrogen, that can mitigate methane and reduce the greenhouse gases effect. They play instrumental role in soil and plant and animal health. They're there to produce our food, and they also support the human health and everything which was done in the human health and linking microbiome, human microbiome with human health in the last years was also amazing. So this is why we have to consider them, but it's also important to start considering them again in this system. I will be constantly telling you system, because they're actually interconnected, and this interconnectedness is something that is not yet so fully explored. So this is from one of our publications that we also published together with different partners from the project. And we try to showcase a lot of these links are sort of hypothesized. There has, to my best knowledge, never been a project that has really followed it through and made all of these connections in one project. But when you just think about it, the microbiomes are in the soil, they will sort of affect the quality of the plants that grow on this soil. And this is something we know. What we also know is that the quality of the grasses, of the food, will also affect the health of animals. And we know it also with humans, these links. So we also know that a certain kind of food that especially cows and other ruminants eat will affect their rumen microbiome and will also affect their sort of methane emission, how they affect this whole climate change. But we also know that, you know, by indirectly affecting the health of the animals, it will affect the quality of the milk, of the product they give, which will again be somehow translated in the quality of the food. There was an excellent study on, like, for example, mozzarella in Italy, and how the microbiome of the initial milk sort of affected the microbiome of the final product of this nice mozzarella that Italians produce. And then we eat this food. And I mean, this food that we eat and the microbiomes in the food that we eat will affect our gut and our health is also sort of clear, but you know, the links are just starting to be established. So we do have a lot of studies showing how different diets affect our health, but this direct sort of transmission of microbiomes from food to gut, this is also just at its very beginning. We're just starting to scratch the surface. But we also then need to think about this circularity, the waste treatment. So like the waste from the animal production, the waste from the food production, how can we reuse them? Not only thinking biofuel, but also thinking upscaling waste, extracting additional, you know, really good and beneficial molecules and somehow returning them into the food chains. And we know that microbiomes can have a plethora of functions that can be used there. So again, a call, whatever you do, think of all of the systems around you and how you can link about them and how what we do actually links to this and affects these other systems. And this is sort of a landscape where Microbiome Support and its consortium started working. And even though our official kickoff was in November 2018 in Canada, actually the first real meeting and the first working meeting was in Vienna in March 2019. And this is a photo I really like because this is how we started. We really started pre-COVID. We started live. Everybody was happy. Nobody was wearing any masks. We had a beautiful weather and quite warm weather in Vienna in March. And we had over 100 participants at the workshop. There were members of the consortium, but also members of our advisory groups and also different representatives from Commission and from the IBF countries. And we had a great days discussing on, first of all, starting to sort of paint what is the status quo, how different stakeholders see the microbiome research landscape, and also a lot of visioning and, you know, how it should look like in several years and what are really important steps that we have to take on this way. And I have to honestly say at the beginning, we were a little bit worried that, you know, we will get completely, you know, different ideas from different groups and that they will all have different needs. And, you know, oh gosh, how are we going to sort of put this everything in one nice box and one nice package? But at the end of these 2 days, we were actually fascinated that almost every group came back with the same message. And these were actually the bottlenecks that were identified by all three groups. So first was lack of collaboration between sectors. And this came from industry, but it also came from science. The scientists said, okay, we would really need to understand industry better. The industry said we need to have better collaboration with science. Accessibility and utilization of research data. Everyone wanted to reuse the data. Everyone wanted to have better access to data. The fragmentation of all the activities. Of course, the policymakers were stressing this out, but then scientists came in and said, yeah, but you know, you're investing so much in human health topics and we need to address all of this primary production and the whole system. The regulation, you know, honestly, we were thinking it's going to be a big issue discussing regulation, but the regulatory people said we need to understand how to really make a good assessment of microbiome-based products. And the scientists and especially industry said we need to have clear regulation for our products. So everyone wanted to have clarity in this area. And then also there are some sort of, those were like main bottlenecks when we were thinking translation of research into the application and into the products. But then everybody was also sort of mentioning, yeah, we need to also improve public awareness. We need to ensure that these products and the microbiome application will be accepted once when we hopefully start entering the market on the big scale. We also need to have a good expectation management. We know that, you know, there are some chemicals that we try to replace with biologicals, and biologicals don't always perform at the same level, but they're still perhaps or definitely better than, you know, when you think about the bad side of the chemicals. So there is also really need to deal with this expectation management, not to raise hopes too high, but just to, be on the same level. And also, okay, put the scientists into the room, but everyone is always going to scream about we need better funding. And, if you throw the line below all of these points, actually, we wanted to have more coherence in the landscape. We wanted everything to be more focused and somehow more aligned to each other. So we as a consortium took that as a confirmation of where we started, and we really started working on that. And I will now show you how we addressed some of these problems and what sort of solutions or recommendations. We are just the consortium, we can only offer recommendations, but I will also show you in which sort of landscape we did that and why we hope that it will really make a difference. So one topic was fragmentation, and everybody was sort of saying, yeah, the microbiome research is fragmented, and people from plant and soil and animal scientists said, help people get more money, but actually, this was all somehow a gut feeling, and we went then set out and did the big mapping of the microbiome research landscape, and this was the effort that was led by our partners at the Wageningen University in Research. But all of this, as you remember at the beginning, I showed you partners and as a national contact points. All of the partners participated, all of the partners collected data in their different countries, the data about what funders we have, what funding programs we have, what projects are getting funded in this area, what policies and strategies do we have, what infrastructure do we have, so what's available in our country. Everything was sent back to us, everything was analyzed, and we established this great database where you can also query all of these things. So this is freely accessible resource. You just go at our website, the link is down there, and you can just have a look at what's happening in different countries. We also categorize the projects in different fields. So you can just search, who is doing what in which country in my area. And we, of course, analyzed the data, and we saw, and I'm now showing the status of 2019, that when we sort of just summed up all of the projects that were tagged as research in the microbiomes important for human health, we had over 2,500 projects reported. When we looked at the projects that were addressing microbiomes of food production and processing, we had something over 400 projects. Now, I'm aware this mapping is not perfect, but it was as good as we could do it, and still the difference was amazing. We did a mapping update just this year, and we are now updating the database, so there is going to be an announcement when the new version comes. There was a slight shift of this trend, but nothing spectacular really happened. The biggest shift that we saw since 2019 and now was that the number of strategies that were really directly addressing microbiome has increased. And this is good because this is actually a good starting point, because the funders look at the strategies and make their funding calls based on the strategies. So this will also help us increase, hopefully, the proportion of the projects. What we also discovered is that a lot of projects are really small projects, like up to 2,050, 300,000 euros, which actually doesn't allow you to implement this systems approach. Most of the projects are enough to have one early stage researcher working on his or her topic. So this is also something where we are also linking this fragmentation of the research back to this need, to how we address the research. And also what we noticed, a lot of the research that has been done in the past is really sort of descriptive and taxonomical. And we know that we need to go more into the direction of functional, but for all of that, you need bigger projects. And to sum all of this up, we have recently published a paper. This is really a paper. This is based on one hand on this mapping exercise, but also on the really in-depth research into all of the published work, because this was done as a desktop study. And we have published this paper. We really call for systems approach, where we say, okay, we need bigger funding, we need bigger projects, we need better collaboration between sectors and internationally, and we really need to implement this systems approach in the research in order to achieve our goals. So it's really an interesting paper, as I said, again, a group effort of the whole consortium. Even some of the members from Brazil were there. So I really hope that you go out and read it and, also disseminate this, because the more people we get to read this stuff, the more people we get to understand this stuff, the better it is for our research landscape. The second big problem was, I already mentioned, this accessibility and utilization of research data. And we have a bunch of workshops addressing different subjects. So we know, is microbiome always used in the same way? So what's the real definition of microbiome? This is the one point where we started, but then we were discussing about the biobanking and data storage, about the whole point of standardization. Do we need it and how do we implement it in this rapidly evolving area? What to do with bioinformatic analysis, how to use existing data, and also thinking further. I said we need to go from taxonomical to functional. What is happening with omics? How do we deal with omics technologies, and how do we integrate different data sets? And this is where what happened. We started really nicely having these live meetings. This was 2019 in Thun. Those were the first 2 workshops that were sort of affiliated with a big common ground workshop. And then COVID hit, and suddenly we had to do everything online. And it was, as I believe it was for all of you, a big adjustment at the beginning. But 4 out of the 6 workshops were then held online. We had great participation of partners, but also of external experts. And we really managed to come to some sort of a consensus conclusion to say, okay, this is the recommendation with which most of us, most of the community can live. And this is something. All of these outputs, or most of them, you can already found out there. So first of all, we had this really nice effort led by Gabi Berg from the Technical University of Graz, and this really great paper, Microbiome Definition Revisited, discussing all of the old concepts and what are the new challenges and how we see microbiome. And I just gave you the key figure from this paper, that we see a microbiome as a community of the all organisms, but within their theater of activity. So linked with all of their functions, everything which is happening in this micro environment. So this is sort of adjusted definition, the old definition of VIBs from, if I'm not mistaken, 1988. But this paper has so much more. It goes really in depth, showing this research field. So I would really recommend you to go there, just download the paper, is open access, and just go through it and see, where we started, where we are, and how we see that this should develop. But then, going a step further, we have now some publications, a lot about the data infrastructure, about the need for biobanks, really microbiome biobanks, not just the strain collections, about the harmonization of metadata, a really important issue if you want to use and reuse the old data, because mostly you have these huge deposits of sequence data, but actually don't know anything about the samples there. You don't have any metadata about the samples, so they're sort of not of use for you. We also discussed a lot about how then to link this sequence data to the real original biological samples, which you would probably need to follow up with some research. So you will find a lot of these thoughts in these publications that are listed here, and a sneak peek. Some of them are still coming, some of them are still in the review process. So as I said, just start following us and you will get a lot of input in the next few months. I also talked a little bit about regulation, and regarding the regulation, we actually had a great workshop in November 2020. This was the effort that was led by Yolanda Sanz at CISC in Spain. And we were discussing on 2 different levels. We were discussing the short-term vision, like what's needed now and what has to be done now to sort of impact the next 10 years. What opportunities do we have, but what are the requirements, and how to ensure the successful future. But then we also sort of wanted to have this long-term visioning and say, okay, what else needs to be done? We all know that regulatory is not developing so fast, that policy is not developing so fast, but what we need to do and how we need to push also these areas to develop so that we can ensure this whole pipeline from idea, from great R&D, from great translational R&D done by industry, really into application, how to lower these hurdles, how not to have to wait in Europe 5 to 7 years to get your biological product registered. I mean, this is, and invest 2 million just into registration. So how to address this issue better, and how actually to do that when we start moving from single strains into more complex products. We were also discussing a lot about funding, and also about this translational funding, this enabling multidisciplinary collaboration and networking. So very often it's difficult for me to, for example, collaborate with any colleagues in Brazil because we just don't have any co-funded calls. And, you know, I need money, you people need money. So if we don't find a way to, you know, get joint projects, we're never going to be able to really collaborate. And we had our colleagues at the DLR in Germany. They were involved in joining in some of the joining program initiatives that are running in Europe, and they made a case study on different funding tools that were applied in the past, especially looking at knowledge hubs and platforms. And they also made some recommendation, and you can download and found also this fact sheet. Everything is open access just on our website. And these were also some, you know, insights, what's good in these tools, how these tools can function. These are sort of a pots where different countries then pay into, and then, you know, researchers from these countries can take money out of. This is also funding that can run in parallel to, you know, other national and international fundings, and also involve more multidisciplinary collaboration. So we also try to see, you know, there what's available, how it could be adjusted. And all of this, you know, on a technical, you know, all of these technical solutions are sort of like easier to publish in a peer review because everyone's interested in this. When I'm talking regulatory and funding, this is perhaps not something you can really publish in your classical scientific peer review, but all of this taken together, so all of the technical aspects, but also all of these regulatory aspects, all of these funding aspects. We also had a workshop on educational needs just recently. So also all of these needs, how do education needs to look in the future? What does industry need? What does academia need? All of this is now coming and influencing the strategic research and innovation agenda. And this is a recommendation from this consortium, but also extended with the expert we did while creating this research agenda. We applied tools like Delphi. We really, you know, announced it broadly. We had a lot of different scientists, not even involved with the project in any way, giving their feedback. So all of these sort of inputs that I showed until now, together with everything which was happening in Delphi approach, is now coming together in the strategic research and innovation agenda. And sneak peek, it should be launched and should be made available within the next few weeks. So this is our big outcome of the project. And I will just show you one little picture of this. It's structured around 9 areas of interest, and also defining these key targets and goals for the future. And talking about these 9 areas, we have we have identified five different thematic areas, going from the agri-food systems, but also involving the animal, but also the human health, the food production, again, food systems approach, just a little bit different, but also including these transversal topics about research infrastructure, about data access and sharing, knowledge transfer, fragmentation, and also this improving stakeholders, but also society awareness and capabilities. I have read the text. It's really a great text. It's not, you know, it's sort of written, also co-written by scientists and policymakers. So it's not your typical document that you just sort of don't get after 2 pages. So I really hope that you can use it and use some elements of this and also promote it in your country, or use it to implement some ideas in your country as well. One part that I haven't touched, but one of the really important activities of these projects was enhancing dialogue between stakeholders. So I showed you we had these different groups, advisory group within our projects, and we always had members involved in our meetings, but we also went out and made, you know, meetings or attended meetings at a different level. And we were present, for example, at European Research and Innovation Days. This was also 2019, as you see from happy smiling faces without masks. But also post-COVID, a lot of online events, and we were really proud that we actually were able to host and organize 2 events together with the European Commission, where actually also the European Commission experts were there, were reporting along for us and were discussing for us. So we had like one event with the European Parliament and one event with the STOA panel. And also these inputs for these events are sort of have influenced the development of SRIA greatly. We also went in this bioeconomy sector and also sort of reveal to the people who are really focusing on bioeconomy what the microbiomes are and how we play also in their system. I was mentioning a lot the IBF, International Bioeconomy Forum, and just to give you also a sneak peek into that, International Bioeconomy Forum is a loose affiliation of different countries around the world. So Commission is represented as, European Union is represented through a Commission, but then you have members from Canada, from US, from Brazil, from Argentina, from South Africa, India, China, Australia, and New Zealand participating, and they're in different working groups. So there are now 5 working groups. The last one, I should have put it in black, apologies for that, is also already active on biorefineries. But there is a working group on the microbiomes in food system, and this is actually the working group that is led by the European Commission. This is where European Commission put the biggest efforts in. And this is where actually mostly policymakers then meet and sort of discuss how we can collaborate internationally, what needs to be done to push this subject further internationally, and how can we really, and as you see from this title, it is a world effort. We can do whatever we want in Europe, but if the Europe is quite small, we are not going to be able to save the planet on our own, and we are not going to be able to feed the planet on our own. So it's really a lot of push in this group to collaborate internationally, and it was really a great opportunity to, as a microbiome support consortium, to also bring a little bit the scientist and the industry voice at this level, because policymakers often see these big goals and they're important, but we sometimes need to inform them that not everything is so easy as writing, oh, we are going to be climate neutral by 2030 or whatever. There's a lot of effort that still needs to be put into coming there. So we did a lot of work together with IBF, especially in the microbiome group, sort of trying to give input to their discussions and considerations, but we also developed and hosted a website. So now you can finally find a lot of information on the IBF, which you couldn't when we started writing this proposal, and also look who is active in these different groups. And also, they're always open for people contacting them, bringing in opinions, bringing in topics, and just trying to establish the collaboration. And the publication, I already showed you one graphic in, it was actually a Nature publication. It was one of the great publications. It was written in scope of this, as you see from the author, is in scope of this IBF. So we had the members from New Zealand and Canada and Australia participating and giving input. And really, we are also showing in this paper a global landscape. So what's happening in different countries and how different countries address this issue. And also, again, finishing this call, how we should do it together to really improve and to really benefit from all of our efforts. Registrations are not open anymore. Sorry for that, but this was the only image I could now find online. Registrations are not open. We just had our final event at the end of June. Our big final conference, happy to say, again, live event. So after 2 years online, we were able to see most of our partners again live. It was a great event. We again had over 100 participants for 20 countries. We had, again, policy, industry, and scientific stakeholders participating. A nice thing was we were in Brussels. It was really intentional because we had a lot of policy representatives from European Union presenting there. You can find many presentations available on the website of the project. So if you want to go and see what different directorates of the European Union think about the microbiome and how they see them in the little area, you can go there and click on different presentations. We were also really proud to have John Bell, who is the sort of director of the Healthy Planet at the European Commission, to give a plenary talk or contribute a plenary talk to this lecture. You can find recording on this online as well. He reflects a lot about this international and also about IBF needs. A really nice short YouTube video you can access to our website. But what was also interesting, we had this panel discussion with representatives of FAO, with representatives from the project, from the science, from the policy. And we were discussing, again, the microbiomes, how can they improve the food system, but also most importantly, how can they help us to address this crisis that they're sort of, we were faced with COVID. We knew that, how COVID affected some people with some illnesses, how these illnesses are also linked with sort of bad nutrition. We also now have in Europe the issue of Ukrainian-Russian war, which is also affecting our food security or threatening our food security for the first time in many years in Europe. And we were also discussing how can we really take this potential of microbiome and use it to sort of also address this sort of non-predicted or like imminent crisis that we are sometimes getting. And I also have to say there is now manuscript being or has already been submitted as a basis from these discussions that again went beyond the project, went with the FAO and you know, again, includes like a global perspective on the microbiomes and what they can do. Improving public awareness and acceptance is a topic really near and dear to my heart. Because I think we also need to make sure there's sort of a lot of communications, a lot of stakeholders are communicating, and what I would really like, I want everyone to make their own decisions if they are for or against something, but I really want this decision to be educated. I want it to be knowledge-based, and that's why I think we really need to make sure that public is aware of what microbiome is, and I think that the gut microbiome, the human microbiome really paved the way from us. But I think there's still, and I know there's still a lot to be done. And also I saw also with some policymakers, I told you that the policy, the sort of representation of microbiomes in policies around the world improved. But I talked with a lot of policymakers at the final conference and they said, oh, that's quite some concept to wrap my head around. So really showing me there's still a lot to be done. So what the microbiome support did, we have a nice website and a Twitter account, and this sounds sort of a newsletter and sounds quite usual, but we really put a lot of effort in there, a lot of effort in communicating. This is not just ticking the C&D box for us. We have also started the microbiome support ambassador campaign, which we're really invited to join, where we ask the people to say, okay, will you be a microbiome support ambassador? And if you support our goals and our visions, why? And we have a bunch of people, again, on our website that were just declaring their support for the microbiomes and explaining why. And there's also on the website sections where you can see all of these nice testimonials, but you can also send us yours and we will be more than happy to upload it on the website. We have also established a close collaboration with something which is called World Microbiome Day. And this is the initiative that was started in Ireland by their APCC Center. And we are now sort of, our colleagues at UFIC are now sort of collaborating with them on that. The World Microbiome Day is on 27th June, and thinking back about our final conference, this is also how we placed it. And this is a day where we sort of call out to everyone out there, to every scientist, to every school, to make a sort of public event. It doesn't have to be on the 27th of June. You can place it somewhere around it, but announce it on the web page and go out, reach out to your community, reach out to your, I don't know, school kids, senior citizens, farmers, make some event, inform them what the microbiomes are, inform them what you are doing, try to show them what our vision is, and try to include them, get also their feedback, what they want, and you know how we can also collaborate with them. And if you don't know how to start that, and I know it's a difficult, and it was also troublesome for me, microbiome support gives you a whole bunch of resources which you can just download and use for free. We just ask you to acknowledge us on the way. We had these infographics I have shown, we have videos, and infographics and videos are now in the last months getting translated on many, many different languages. So I think that, you know, your language is already there, but if not, reach out to us or to Professor Arruda to come out to us. We can still squeeze in some translations if needed and have these resources at your national language for you to use and to be, you know, we are aware. I mean, I work in Austria. It's difficult to take an English flyer and go to talk to Austrian citizen, but I have it in German, which makes my life quite easier. We also developed a coloring booklet for kids. This is currently only available on English and German. But as I said, you know, it's not such a difficult text to translate, and we can then help you also put it in form if you need it. We have all of our fact sheets. We had the microbiome research database I was addressing. We have some podcasts, and a nice thing is also together with other projects, which you will see quite soon, we have now also developed a sort of microbiomes for life communication campaign, and there's also a huge, quite a large set of social media material that you can use. And we just ask you to acknowledge us. So all of this is free for you to use, and we hope you use it because we really wanted to have a broad application. I will also give you a sneak peek, and you're really first ones to know about it because we just 2 days ago got a letter from Environmental Microbiome confirming that our publication has been accepted for publication. And this was an effort that was led by Lene Lange from Denmark. She's now sort of working as a private consultant, but she's a former professor, and she has a long track record in the industry and is actually an amazing lady. And she really led this effort and said, people, we need to talk about ethics in the research. We need to talk about ethics and how we deal with our resources and how we deal with our knowledge and how we manage this globally. And I'm really happy that she was behind it, and it's a great publication. And I don't know, it's probably going to take some weeks until we are online, but it's coming in Environmental Microbiome. So please keep your eyes open and read it. It's really an important piece of work addressing really, really important issue. And we have few more publications in the pipeline, some of them dealing with omics and educational issues, and some on a more global level on this, further advocating for the systems approach. To sum up, we have this vision, and sort of what we also said, we have now this sort of a pathway that we envision of how to come from this research and innovation and go into the impact. And this is now just a summary, and I'm not going to read it, just a summary of many points which I have addressed. We are going to sum this up into 3. We have elements of this in our different publications and fact sheets and other materials. So this is our vision, and I hope that we have now put enough noise around it, that we have sort of communicated it enough in the past 4 years, that we have discussed with enough important stakeholders to make sure that we are going to start to move along this way and really go from this great research and development we have had in the past years and really start making some impact, really not only entering the market, but making impact for the society, for the planet, for our environment. We are also already, and I don't want you to have the feeling that I think that we are just doing a great research. We already have a success stories, and this is also one of the publications. We have made a selection of case studies, and you see again, food systems approach. We have 14 case studies summarized in this great review that was effort led by Rocio and Martin Wagner from the Foxy Center in Austria, but again, collaboration of many different partners. And we have these 14 case studies. These are already applications, products or methods that are out there that are being used, spanning plant health and feed products and livestock health, food production, but also human health. So if you want to see how these things went from great R&D idea and a great peer-reviewed publication in a scientific journal, and how they really went into the application, I strongly recommend you go there, pick your favorite story, and as always, feel free to give us feedback. What do you think? And I hope you also find some really inspiration and motivation in this paper as well. Also, there was, in parallel to the microbiome support, there was some spectacular research being done, because exactly parallel to us, there were 4 big innovation actions. So really research projects running called Circles, Hollow Food Master, and Simba. All of them were approximately 10-11 million euros, so big projects addressing these microbiomes in food systems, each one of them in their own way. I have here links for you. I will not present now all 4 of these projects, don't worry. But go in there, look what kind of food, plant or animal-based food they were using, what other efforts they were doing. All of them are now at the end, also ending, but you can still reach out to the researchers and connect with them and see what was published and how you can follow up. And these 5 projects together have this Microbiomes for Life communication campaign. This is something that we are running together as a joint effort. I was now talking a lot about the microbiomes and about this framework, but I would also like to just give you I'm a scientist at heart. I was just going to give you a little bit also of the insight into some of the research that we are doing. And for this, I'm going to now switch gears and go into the master projects. Project Master stands for Microbiome Applications for Sustainable Food System Through Technologies and Enterprise. It is one of these innovation actions, which is coordinated by Paul Cotter at Chagas Institute in Ireland. Master is the project that really applies this food systems approach. As you can see from this image on the left side, we address different parts of the food system. We address the marine and the fish production. We, of course, address plant and soil, focusing on fodder grasses and fodder maize. There's also a work package addressing animal health and how feed affects their health and productivity. There's also something about this, what's often disregarded, thinking about waste and how we can upscale waste. We are in the food production facilities, looking how we can use microbiome also for source tracing, for pathogen identification, for food production, and also looking a little bit into this aspect of human health. So going all in every sort of little place where microbiomes can play a role. But we also have this sort of transversional topics looking into what are the services, what are the methods, what are the databases, how can we connect and share this data, sort of underpinning our own research within the project, but also underpinning the society that can use our project. We have a large set of aimed outcomes going from, touching all of these points. And as you can imagine, this is not something that you can do on a small scale. So this is a big consortium of almost 30 partners from across the Europe. And we don't only have scientific institutions or universities and research centers. We have many partners from industries, from different industries, sort of collaborating with us, exchanging with us, and sort of also you will see also developing products with us, so really pushing it where we need it to be, not only, and don't please get me wrong, the basic research is important. Scientific publications are essential. We need this knowledge to build on that, but we also need to make sure to make this jump and transition into the application. So all of these parts are equally important, but in this project, we really have this nice landscape where we can work directly with industry to make sure that this happens. I'm only going to give you a sneak peek in the work package on the plant and soil microbiomes. I have told you in the core of this work package are two model crops. So we are working with fodder maize and fodder grasses because, linking to our animal work package. And this is the list of the partners that are involved. So again, Angela says, which is together with me, we are coordinating this work package. And then we have colleagues from Chagas. They're working mostly on the fodder grasses, which I'm not going to go into today. But then we have companies like Enoch, a small company in Germany working on IMF, and I'm going to present a lot of that work. We have a Christian Hansen participating with their strains and their biologicals. We have also Anna Diak, who was instrumental in performing a lot of field work that we are doing in this project, and also in this diagnostic work we were collaborating with Quagen. So you see a lot of companies already within this work package. And we were trying to address different topics. So first of all, further development of microorganisms and application solutions, but not only developing an application, but also looking at what management practices we need to ensure that this application is really delivering. We are also looking in diagnostics and prediction tools. So can we really predict what's going to happen? Sort of, my vision, can a farmer make a test on his farm and decide this is the right biological for my system? This is what I should apply to ensure that my system is more productive next year, because we are all aware there's no one fits all solution. So we were addressing all of these issues. Of course, our direct aim was to ensure more sustainable production and healthier feed. But if you think further, healthy feed, as I already said, makes for healthy animals, makes for healthy food, and makes for healthy people. So, you start there, but you need to think further. You need to think about these other systems. And I'm going to just talk about what was done in this development of microorganisms, application and management practices. And I'm just going to address a little bit the work that was done on the maize and with mycorrhiza and some bacteria. And I want to really start by acknowledging also my colleagues from ENOC who provided a lot of these slides and who have done most of this work. I'm just going to brag around a little bit because it was great results. And I will just give you two brief slides about the mycorrhiza. Perhaps most of you know it. The mycorrhizals are obligate plant symbionts, and there are different kinds of fungi. They're sort of classified in different groups depending on how they interact with fungal roots. Do they only associate or do they really sort of grow into the root? And you will also see that the biggest group are so-called arbuscular mycorrhizal fungi, the IMF, as I'm going to refer to them, and they sort of co-colonize with 75% of the plants on the planet. So it's really, as you can see, an important sort of symbiote of plant that we can use. This is a life cycle of the IMF. So as I already said, they're obligate biotrophs. They need a interaction with the plant to sort of be able to fulfill their living cycles. If they sort of exit the plant, they will enter the dormancy state. So for their active part of the lifetime is symbiotic and sort of happens together with the plant. And they have many positive effects. Some of them are direct effects like biofertilization. If you can imagine the root with this nice AMF hypha around it, can definitely sort of get more nutrients to the plants. It's also sort of a bioprotection because you already have this niche is occupied. So, you know, the pathogens or whoever wants to come first has to deal with AMF. They also sort of have a bioregulation effect on the plant because also they communicate with the plants through exchange of different signaling molecules. They play again a role in the structuration of microbial communities around the root. So they also choose their partners in the rhizosphere. And also, since we are thinking AMF, we are thinking these hyphal networks, they do a lot to structure the soil, to make it more, you know, to enhance these pores that then again are for better nutrient and water retention and exchange. So they really play an important role in the plant life cycle. And the aim was to develop like better production methods and better formulation for already existing AMF fungi and to test them under field conditions. And also one idea was, let's try to combine really the AMF fungi and bacterial treatments to even further improve the plant biomass and quality. So as I said, the aim of this project is already on a quite high technological readiness level. So we didn't go out, we didn't sort of isolate and test. We took the strains with the known beneficial properties that have been shown in previous studies. Actually, this fungi was already available as a product with Enox. The bacterial strains came from AIT and Christian Hansen, but we had a lot of data showing that they are sort of biofertilizers. And then we sort of went out, especially partners in Enox, and wanted to see the inoculation effects, the concentration of what needs to be applied, how it needs to be applied, how can we apply them in this combination of AMF and bacteria, what management practices are needed, so also formulation development, but also what is the fertilization effect and how to optimize the system and what recommendations to give to a farmer. And I will just give you some of the results here. So first of all, we had 4 different microorganisms that we test with this IMF, and we just sort of did this sort of you know, how they deal with each other, sort of a small test. And I apologize for not showing any specific names or data, but we are just still in our publication and in you know our market launch phases. So I'm not at liberty to do that, but I'm showing different colors, and I think you will see effect. So what we did here, we have tested the frequency of colonization, so sort of the number of plants that get colonized and the intensity. So how many propagules did we see at the plant? So those are the 2 different graphs, the F and M graph. And they tested the 3 different concentrations of AMF. The first bar in the light blue is sort of the AMF control, the fungi inoculated alone. And then we had these 4 different treatments, and I will just highlight 2 of them, the orange and the dark blue, so the second and the last one. And both of them were bacillus. This is all I'm at liberty to say, but interestingly, one improved the colonization of AMF and also sort of concentration dependent, but the other one had a negative effect on colonization. And this is something that we also got curious, and we are trying to follow this up. I still don't have final explanation for you, but I can only say this is also something that we are now trying to go further into, because we also hope, back to basic science, understanding what's happening will help us further when we sort of develop further projects or try to co-develop these solutions. But definitely, the mycorrhiza colonization was affected by bacterial strains. These effects were concentration dependent, but also dependent on how good the AMF liked or disliked the bacteria involved. Then also, the colleagues looked at different fertilization strategies, and they tested here on different soils and additions of different high phosphorus and high nitrogen sort of fertilizations. There's many things going on to this graph. Just focus on the last 3 bars that are rounded, and you will see here that actually it's always nitrogen that improves the colonization. It's always nitrogen that improves, that also helps that after sort of colonization has happened, that also the plant biomass was successfully increased. And this is what, at the end of the day, we would like, not only colonization, but effect on the plant. So in the first field trial, it was okay, it's clearly nitrogen, but not phosphorus. But then we went in even deeper, and many different nitrogen fertilizers were tested, sort of ammonium nitrate and only ammonium and only nitrate. And actually what we also discovered, that the best effect was always achieved when we were applying the nitrate fertilization, that this was the most compatible with AMF. And this is really important when you sell your biological to the farmer, to also tell him, yeah, but take care what you're using together with it, because, inadvertently, by using something that's not so beneficial, they will not see the effects, and then they will sort of think, oh, all biologicals are not working and it's not so good because, just the conditions were not fitting. So also important always to see and to give recommendations on how to use your biologicals. It's not chemistry, they're a little bit more sensitive. And then as a last part, I want to show you the results from this year, and this was the formulation. So as you can see on this little picture, I mean, this is sort of a crude formulation. This is how the colleagues were applying mycorrhiza before, and they were just sort of applying it directly into the furrows, and they wanted to have something more stable, more protected. And this is why in scope of this project, this pellet formulation was developed. And in this trial, we tried so different. So there was a non-inoculated control, and then there was AMF applied like in standard form in the crude formulation. And then there was a pellet formulation of sort of only AMF, only bacteria. And this is the beneficial, the one that AMF liked, of course, and a co-formulated pellet of AMF and the bacillus. And then there were also, again, 3 different fertilizer regimes applied. One was the standard fertilizer that's usually applied in Europe, or at least in this region of Europe, in Germany. And then also, since we knew that the nitrate is the one that they prefer, we tried 2 different nitrate fertilization regimes. And it was quite interesting. So you always have, this sort of different biological applications always across these 3 fertilization regimes. And interesting was that the biggest difference was actually observed under the classical fertilization. But what you also see that we had a big yield increase, and this is really a significant yield increase of almost 40% with AMF pellets, but also the bacillus pellets performed quite well, and combined pellets, like they didn't have the add-on value. Not in this study, and this is what you see, you have 2 beneficials, you add them together, and still we obviously don't have these conditions perfectly sorted out, how we can sort of manage these 2 together and make them collaborate even better. But all of these treatments under this one fertilization condition really improved the plant yield. And not only that, and this is now where it becomes really interesting, that these treatments actually also improved the cob filling. So sometimes you have these cobs where tip of the cob is empty, and what we have seen with applying these different biologicals, we had significantly better cob filling. Again, AMF was the best, but also Bacillus had significant improvement of the cob filling, which is great news for the farmers. And this also resulted in significantly higher number of seeds per plant and bigger seed mass. And what we have also seen, actually, that we had bigger seeds when we were treated, and sort of the seeds themselves, sort of that we had bigger yield, and the seeds themselves were bigger. And we said there was absolutely no effect to the grain quality. So we didn't, there was some quality parameter test. There was absolutely, so it wasn't like, that we lost on quality. We really improved the yield without losing a quality. And what we are really proud of, there is a thing called EU Innovation Radar, and it's a platform of the European Union that sort of screens European projects and sort of highlights the innovations that are really ready to make this jump into the applications. And we had several innovations from master that got selected for this innovation radar, and the one that I now presented is also up there. The link is not really good. I will try to send you a better one. But if you just go on the innovation radar and search with this number, you can get down a lot more information about this new pellet formulation, and also how the European Union sees the chances of going into application with that one. You're also more than I would be more than happy to share the contact data from my colleague. You can also contact them directly if you're really interested in this AMF work and want to connect with them. I'm now at the end of my presentation, and I just want to finish with a few acknowledgements. And first and foremost, I want to really thank both of the consortium and all of the consortium partners, because all of this is always a group effort, especially in the microbiome support, but also in the master. And when you have a good colleagues to collaborate, then you can really, move faster to your aim. I also want to acknowledge my boss, Angela Sesic, who is also microbiome support coordinator, but who also, made for us as a team at AIT this, sort of possibility to be present in this landscape. She did a great pioneer work in this endophyte, and this is why we also get invited and get to participate in all of these great projects and collaborate with all of these amazing people across the Europe and world. I would also especially want to thank my colleague Eva Lucic at INOC, who is responsible for all of this great research work I was showing. And of course, no work can be done without a proper funding. So I was always acknowledging, at the bottom of my slides, the two EU projects. Here again, the two grant agreements, one from the microbiome support and the other one for master. And really a great appreciation for European Commission and all of the funding they put into the science, because honestly, I wouldn't be here today with you, and I wouldn't have anything to talk about without them supporting us now in the past. And with that, I also want to thank you again for invitation and for your attention. I hope you found something interesting in my presentation, and I'm now more than happy to take questions if there are any. So thank you all. I think I will stop sharing now, because then I might be able to see you.
Thank you, Tnaja, for the very nice presentation with the overview of the output of the microbiome support project. That was fantastic, and we are very happy to be one of the members of the consortium. And indeed, Rafael, that was researcher, our colleagues that participate most on the activities of the project. So thank you very much. And I open for question. I don't know if anybody here in the audience has a question. So, Rafael, please. Come down.
Hello, Tanya.
Hi, Rafa. Nice to see you.
Congratulations for the presentation. I have a question regarding the last slides in which you showed about the mycorrhiza. I was curious to know a little bit more about the challenges regarding the formulation, because as you said, in the case of mycorrhiza, they are obligate symbiotics. So you need to have the plant in association. How long did it take so you could make the formulation for those fungi?
I mean, I cannot tell you any details about this formulation, but it was like a 2-year effort led by Inox and some of their other partners. And if I'm not quite mistaken, I think this was already like a 10th version that finally worked out for them. So it was a lot of work put into that, of course, to make it stable. You know, they have to produce it, as you say, and then to stabilize it enough to be able to formulate it, not to kill it during the formulation projects, to sort of keep up the viability. So it was a long process, and they were really quite happy with the results. So after the first year trial, they were like, these things are like sort of almost too good to be true. Let's go for the second year. But we got, as you have seen, even better results. I mean, we are aware that we are now mostly limited our testing to Germany, which is one specific climate. And we would definitely also like to extend it, but they really plan to launch this as a product towards the end of this year. So they are quite confident that it's working really well for them.
So I have a question, Tanya, regarding the regulatory issues of the microbiome. I know that European Union is, it's not so easy to get a consensus of all the countries participating in this. I can imagine, But a question is, that Brazil is, we have a very interesting, let's say, I think the best case for soybean with rhizobium for nitrogen fixation in soybean, and Brazil is the largest producer of soybean in the world. So there's a soybean from Brazil goes for every country maybe in the world. And the problem is how you think that regulatory issues could in the future, because more and more, let's say, microbes are, being studied to put in the market as inoculant or any other application. How you think in the future maybe regulatory issues could, anyway, prevent, utilization of this technology in large scales like for soybean?
It's a huge hurdle. It's still a huge hurdle because, as I said, in Europe, it's also it takes a long time to register a project. And I was discussing with one of the members of the European Commission, and he said, well, like, we fast-tracked the project. It was 4 years registration process. It's fast. And I was like, for a company who put, a lot of money in R&D and they cannot start making revenues until they can start placing the money. And 4 years is forever. It's not fast. It's not fast, in my opinion. So one problem is that they need to streamline these projects, and there are now some efforts in Europe to really do so, to really shorten it. And I mean, I don't want to make any false hopes. It won't be 2 weeks next year, but I hope that we will really significantly reduce it. Another point is that we have, unfortunately in Europe, we have European regulations, but then still every country can put something on top. I don't think this will change, but I think what really changes strongly is that there are a lot of scientists at EFSA, at European Food and Safety Authority, that is they're really aware that current regulations or, current requirements, the things that you should show to register the projects. I mean, unfortunately, they more or less took the chemical guidelines and translated it once to biological. Doesn't work. I mean, I don't need to explain you that. But they are now really sort of pushing for this joint effort with the scientists. They're making a lot of also public consultations in Europe. I mean, those are involving more scientists to sort of co-develop the new regulatory lines. to really make them more suitable for biologicals, but not only, and I think this is going to also be a huge hurdle, not only single strains, this is manageable today if you have enough patience and money, but also what happens when you start applying syncoms? You know, this is the next level. What if you want to mix 3 mycorrhizal fungi with 1 bacterium? What if we have 4 organisms? And you know, what if we even go more complex? So this is the biggest hurdle, and this is what we tried telling them, and what we were telling them for 4 years. You know, you put a lot of money in R&D, and you ask yourselves why there are no applications. We cannot go into commercialization. The regulatory hurdle is too big. You need to think there. And I think we screamed loud enough in the last 4 years, and I see something happening. I see also the European sort of for biologicals, they're now completely revising this. They really want to fasten the process. They have realized that. But yet, you know, it's still not harmonized with the regulation in Brazil. It's probably not harmonized with the regulation in Canada or in, I don't know, somewhere in Asia. And there's still a lot of work to be done. But I think it's also a lot of responsibility of scientists to go proactive on that and not just sit and wait, see what, you know, the regulatory people do, but go there and tell them this needs to be done and you need to consider these points. And these points are important, and go to talk also to your colleagues internationally, because it's not just the Brazil, you know, Brazil is also not going to be able to solve alone the problem of world hunger, and Europe on its own, you know, I think we are altogether smaller than Brazil. So we need to really push more international considerations there as well. But it's a huge hurdle, and the only thing you can do against it is be proactive. Be proactive, go to these people and start communicating with them, start educating them, give them the knowledge to understand why the system is not working. I mean, what we realize, they're also trying to do their best. They're not trying to hinder us, you know, they know how chemicals were registered, so why not do it for biologicals? So go out there, educate them, tell them why the system is not working. Tell them that you have problems to, you know, apply your rhizobium in Europe. Tell them to talk with their colleagues. I mean, they also collaborate on certain levels. Push them, be boring. This is the only way I think we can make a significant progress.
Okay, so thank you, Tnaja, for yes. That's we need to. It's like the other biotechnology tool. So we need to educate legislators, and it's not easy. But communication is again very important, and I hope that for the microbiome applications should be a little easier than, for example, for other biotechnology tools, you know, for improving food production. So if you don't have any other questions, so I thank you very much again, Tnaja, for the nice talk, giving this nice overview of the actions of the microbiome support, and thank you again. And I hope next year we give this, we make this event every year, and I hope next year you can have opportunity to come to Brazil to visit us.
Well, it's on my bucket list, so I would love to.
Okay, so thank you very much, and have a nice day.
Yeah, have a nice workshop, and thank you for the opportunity. Bye, everyone.
Thank you. Bye-bye.
Bye.
Agora a hora do almoço, às 13h30. Às 13h30 nós vamos ter mais microbiome. Agora com um pouquinho mais envolvendo a questão da como é que você avalia, como é que você descobre realmente se um micro-organismo pode ser isolado de uma planta, pode ser aplicado em outra e assim por diante. Como que a gente analisa tudo isso, né?